Our next presenter is Dr. William Kisseberth, who is Associate Professor in the Department of Veterinary Clinical Sciences at the Ohio State University College of Veterinary Medicine. He earned his DVM from the University of Illinois and his PhD at the University of Wisconsin. And he works very closely with the Veterinary Medicine Biospecimen Repository, which he will talk to us about this morning. Thank you.
Okay, thank you. So thank you for all being here. I guess I could start by saying now for something completely different, but part of the message I want to give is that a lot of what we do isn't a whole lot different than what Nilsa talked about, with apologies, though, that when we went over and visited her a few years ago to get some advice, that she did not suggest that we change our name by the definitions that she just gave you. I guess we would be more, perhaps more appropriately referred to as a tissue bank. So as you can see, there's 3 to 4 people in our operation, but a lot of what I'm going to talk about is really a little bit about what we do, about the dog model and what it can offer to at least some of you, and the types of tissues and other biospecimens that we have available. For those of you who aren't that familiar, we're located over the river, the veterinary school. Ohio State likes to tout itself as being the most comprehensive biomedical community in the country, and that the veterinary school is one component of that. We are one of the older veterinary colleges in the United States. Building on the left is the Veterinary Medical Academic Building, and on the right is the hospital where we're located. We're in the just broke ground last month as far as a major renovation to the hospital. Whether it will really look that good when they're done, I'm not sure, but we're hopeful. So why dogs? Why might you want to think about the dogs in your research? I'm a veterinary oncologist, so I'm going to speak mostly from an oncology perspective. But at least for genetic reasons, the dogs offer a lot of advantages for a wide variety of genetic and genomic studies that go way beyond cancer. And I'm not a geneticist. Dabble and collaborate some with some of the larger comparative genomics groups, though, across the country, and we've done some studies here internally. The reason that, or one of the main reasons that the dog is such an attractive model for some of these studies is that we have a very highly inbred population that sort of has these subpopulations defined as breeds, and that the different phenotypes of the different breeds vary pretty dramatically, just with respect to body structure, but also with respect to their incidence of various disease conditions, including cancer. And for that reason, some of the different genetic and genomic approaches are much more powerful in this highly inbred population. in particular, for example, for genome-wide association studies that you can get by with much smaller populations on the order of about 100 or so affected individuals to study a particular trait. The other reason is humans are more similar to dogs than they are to mice. Human proteins are both at a protein level and a DNA level. So from that perspective, you can argue that the dog is an attractive model. Spontaneous cancer in dogs, dogs, there are a lot of them out there, and they share our own environment for the most part, and they develop cancer with a very high incidence as well. That about 50% of older dogs will eventually die of cancer. Many of the cancers are similar to those that occur in people. Particularly good examples are lymphoma and osteosarcoma. And for some of these cancers, we see them with a much greater incidence. Osteosarcoma is at least 10 times more frequent in dogs than in people. That's probably actually an underestimation given the number of dogs that we seem to see through the clinic. So we have the ability, beyond just sort of genetic genomic studies, to do translational clinical research, for example, in osteosarcoma, where we're able to conduct clinical studies much more rapidly in the dog than you can do in people for a very rare cancer such as osteosarcoma. And owners are increasingly interested in more advanced therapeutics for their pets. And owners are very motivated when we start talking here specifically as far as tissue banking, that it is very unusual. I'd say probably about a handful of people over the years have declined to consent for tissue banking of their pet's tumor at the time of surgery. So we get well over 90% of the dogs that go for oncologic surgery at our institution will get consented and have their tumors deposited within our tissue bank. So this sort of cartoon just sort of illustrates kind of at the top here sort of this traditional sort of paradigm or approach to drug development, that we sort of have these preclinical models in mice, laboratory dogs, and non-human primates that eventually may have a drug or device that then goes into human phase 1 trial, phase 2, and ultimately phase 3. But by doing clinical studies in dogs, we can actually provide quite a bit of information at pretty much every stage of drug development. In particular, here at Ohio State, we have a clinical trials program at the veterinary school that works very closely with a number of investigators here in the Comprehensive Cancer Center in development, in early phase development of new targeted therapeutics, both new drugs being developed by OSU investigators, as well as drugs that come to us from pharma, and that these clinical trials in dogs can help inform both the both phase 1 and phase 2 study development, that the dog tends to be very predictive with respect, for example, for toxicities, can be very helpful as far as teaching us how to properly schedule these drugs. But in the case of particularly targeted therapies, when we want to start thinking about, okay, how good of a model is the dog going to be for the human, this is where studies using tissues within the tissue bank can be very helpful, both for the aspect of perhaps looking at gene expression profiles in a particular tumor to get an idea how similar or not the dog disease is to the human, or perhaps looking specifically at a particular target to see if it's there in the dog, and I'll show you several examples of these as we go. So our biospecimen repository was established in 2007, initially by Dr. Rush, one of our past pathologists here at OSU that some of you may know. About the same time that it was established, there was a national initiative to develop what came to be known as the Canine Comparative Oncology Genomics Consortium, where one of the initial efforts of that consortium was to develop a nationwide biospecimen resource for some of the more common cancers in dogs that were of particular relevance to studying human cancers. This biospecimen repository were located in the basement of the Veterinary Medical Center, that building that I just showed you. And currently it consists of myself, Dr. Borghese, who's sitting over there. I won't put the laser in her eye, and one technician. Nelsa talked about sort of relationships between biospecimen repositories and how they can integrate into clinical trial operations, and that's very much true with our repository as well. That we're actually housed in the same portion of the VMC as the clinical trials office, and in fact, several of our personnel are cross-trained for work in both areas. So the CCOGC, which is part of the network that we're part of, currently has collection sites for canine tumors in 6 different locations associated with veterinary schools across the country. Those are the yellow stars. And then all the places where there are yellow stars, as well as the red stars, are institutions that are part of an NCI-sponsored network of veterinary academic centers that conduct multi-institutional trials using client-owned dogs. This talked about funding. The CCOGC was established as a nonprofit. Its initial funding comes from several foundations: the Morris Animal Foundation, which is the largest companion animal foundation in the country, the American Kennel Club, and then Pfizer was a primary sponsor of the CCOGC banking efforts and provided major funding. OSU was one of the first 4 collection sites, and as I said, currently there were 6 active sites for collection. The CCOGC established SOPs for collection, and we continue to use those SOPs. So our tissue bank basically parallel collects for the CCOGC as well as for our own internal uses, following for the most part the SOPs of the CCOGC. The major difference in those SOPs is that CCOGC is very specific, that we're only allowed to collect from patients where we are able to get a complete biologic set of specimens, meaning tumor, normal tissue, blood, serum, and urine. We will collect from appropriate patients, even if, for example, we're not able to get a urine sample or are not able to, for whatever reason. For example, with very small dogs, we can be limited about how much blood we can take. So we'll do, I guess for lack of a better term, partial collections on some of our patients. CCOGC is also strictly canine. We collect specimens from cats and occasionally other species as well within the hospital. When CCOGC was established, like I said, the focus was really on some of the more common histologies that are shared between people and dogs. So the first three tumor types were lymphoma, osteosarcoma, and melanoma that were prospectively collected. After they sort of got our feet off the ground, they added a few other specific histologies, kind of a combination of a couple tumors that again have pretty strong parallels in the human arena, lung cancer and soft tissue sarcomas, and then hemangiosarcoma and mast cell tumors, which are two histologies that directly themselves aren't so common in people, but for other aspects of biology were deemed to be of interest. So our SOPs are written down, and we sort of follow this sample processing flow chart with every collection. Our patients are identified before they even go into surgery as potential candidates for collection. They're consented prior to surgery. That once a mass has been removed from a dog or a cat, it's collected and delivered directly to the Biospecimen repository preparation lab to do the collections. We have dedicated staff both within the clinical trials group as well as within the Biospecimen repository for collecting the blood and urine from all the patients, and they are processed here thusly. So just a few pictures from a collection of one of our more common collections. Like I said, was osteosarcoma in the dog. Is probably the histology that we have the most interest and demand for as far as tumor specimens go. That disease is typically treated with limb amputation, followed by some form of adjuvant chemotherapy. So the specimen is delivered to the laboratory, and then collected by the staff for processing. Of course, always taking care that we respect during the course of collection of surgical margins, and that the specimen is able to then be sent on to pathology for proper assessment of pathology with respect to surgical margins and diagnosis. Tissues are processed, aliquoted into, we have collect both snap frozen, embedded directly into OCT, and then also fixed tissue and in formalin for further processing, and then blood is processed as whole blood, serum, and plasma. And specimens are stored as appropriate in liquid nitrogen and minus 80 freezers within the laboratory. So these are basically the collections through the years of these are tumors / numbers of patients collected since the bank was established. Like I said, a subset of these that meet the specific collection criteria, the specific tumor types that were of interest to CCOGC are also sent to that central repository. Just an idea of the numbers of samples in the bank since we've been established. We've collected from almost 1,500 patients representing over 100 breeds that we collect both primary and metastatic tumors, and when possible, we'll collect primary and metastatic pairs. Couple years ago, we became more aggressive in trying to establish more of not exactly a warm autopsy program in that it's a complete autopsy, but we do a warm euthanasia tissue collection where we basically the tissue bank will cover the costs of euthanasia for pets that need to be euthanized due to advanced disease, and do a tissue collection at the immediately after euthanasia, and that in particular has been helpful as far as being able to acquire more metastatic tumors, and particularly primary and metastatic pairs. When I first used the microarray resource here at OSU about 10 years ago or so, and we ran our first affymetrix experiment using dog lymphoma samples, one of the first comments I got was the Corps being very impressed with sort of the quality of our RNA and how clean it was compared to most of the clinical studies that they were used to processing, which I expect was probably a factor of two things. One, at that time, a lot of the early types of affymetric studies were using those types of tissue banks that were just talked about, of stuff that had been squirreled away by various investigators in perhaps not the most controlled manner. And two, that I think where we particularly excel in our collections is in being able to get tissue into into the freezer from the time of removal from the patient, be it surgery or autopsy, of being able to get it processed very, very rapidly. That our technicians are basically there as soon as the tissue's off, and it's being processed within minutes. So we've been very fortunate of having really good results with respect to the downstream applications with the samples collected through our repository. Just off of one of our spreadsheets of just, again, sort of summarizing that we have all sorts of different tissues and from different locations within the bank. Again, CCOGC collects from a handful of specific histologies. We collect pretty much any tumor that is coming off of a patient, regardless of breed or histology. So for even relatively rare tumors or locations that we may have something in the bank of interest. For example, I think about a year ago or so, we received an email from an investigator. I think it was at University of Georgia or somewhere like that, that was specifically interested in colon tumors from dogs. Colonic tumors in dogs are actually pretty rare compared to people, but they were very interested in from a comparative aspect, and although we only had a handful of them, we were able to provide those specimens to that investigator. And likewise, CCOGC doesn't collect from cats, so our hospital overall is primarily, we probably see 75 to 80% dogs compared to cats, but we do collect from them as well. So just a few examples of how samples from our tissue bank have been used over the years. This is example, a recent example out of the Broad Institute of a genome-wide association study for osteosarcoma. And we actually ended up providing the bulk of the Greyhounds that were used in this analysis. The reason for that is we had until recently had an investigator in the college that was really focused on Greyhound medicine. So we had a lot of Greyhounds referred to us. And Greyhounds have the highest risk of osteosarcoma of any breed. So Greyhounds were one of the breeds of focus for the study, as well as a genetically related breed, Irish Wolfhounds and Rottweilers. In this genome-wide association study, they identified several top candidates or loci for further investigation. What's particularly interesting or noteworthy is that the hits that they identified in this study, at least computationally, were thought to account for 57, 85%, and 53% respectively for the different breeds of explaining the heritability of the disease in those dogs. That's very high compared to any, most any type of similar human study. So there are genome-wide associations studies being done in a number of different canine cancers across the world right now, trying to sort of leverage the dog genome for understanding those diseases in humans. Osteosarcoma samples from our tumor bank have also been used in CGH studies. Again, an example from osteosarcoma. Basically, this cartoon showing genomic gains and losses across the genome, and basically illustrating that osteosarcoma in dogs, just like in people, is one of the most genomically unstable cancers, and is kind of a mess to try to study. But that again, that the dog is a very good model. Another thing that we're able to do is to do prospective collections within the tumor bank, either directly or in conjunction with the clinical trials office. And over the years, some people have used samples, fresh samples collected during the course of our collection to develop new cell lines. One of those we actually did, that we established a new canine lymphoma cell line that lymphoma in people, or lymphoma in dogs is very similar to that in people, and establishing new cell lines in dog lymphoma has historically been very difficult, again, just like in people, that it can be pretty challenging, but we were able to develop a cell line and basically characterize it, again, from a comparative aspect. And as well, we've got the primary tumors from which the cell line was derived, so we can then look at be it gene expression profiles or whatever, of okay, how similar or not does this cell line recapitulate the tumor in the original dog, and then we can also look at the gene expression or what have you in that individual dog, and compare it to other patient samples within the bank, and this cell line has actually become one of the sort of workhorse cell lines in canine lymphoma research. This study is, sort of addresses what I was talking about earlier, about okay, we've got a program right now that myself and Dr. London are involved with in the clinical trials office at the vet school, that's been focused on STAT3 inhibition. Okay, well, how good a model is the dog as a model for looking at STAT3 inhibition as a therapy for cancer. So we were able to do various studies looking at STAT3 activation in a variety of canine and human cell lines, and then eventually we're able to go to the tumor bank, remove some primary canine tumor samples from the tumor bank, and get an idea of just how prevalent STAT3 activation is in canine osteosarcoma. providing us with some evidence and further reassurance of the suitability of the canine model for moving forward with respect to clinical trials in this disease. I think that's my, yeah. So I hope I gave you a little bit of a flavor of the dog as a comparative model, the resources that we have over at the veterinary school. Like I said, I wear a couple different hats over there. That I'm one of the oncologists and also directors of the tissue bank. Dr. Borghese, who's sitting over there in the third row, is the lab manager for the bank that you could contact either of us with any questions or requests with respect to the bank.
So you talked a lot about, obviously, the strength of the bank in terms of looking at primary tumors in companion animals. What about for tissue for benign disease? So, for example, feline bladder for interstitial cystitis, or, both equine and dog OA or other things like that. Is there actually tissue banking for that, or plans for the tissue bank to expand? And if so, how does one go about accessing or developing those relationships?
Yeah, I think it's been brought up a couple times. In fact, I think as far as the equine and OA that you brought up, you're probably thinking of some of Dr. Bertone's work, and have talked to her in the past about that. We're certainly have those facilities available at whatever point they want to take advantage of them. I think that again, because of our size and our integration, or close proximity to the clinical trials operation, that it really lends us ourselves to sort of hybrid requests to do various things. The normal tissue question is actually one that has been brought up a couple times, and actually have a proposal on our hospital director's lap right now as far as at least for our fluid samples of being able to establish a bank of healthy normal controls with respect to that. We've also, again, on a prospective basis, for example, have been able to collect. We've had investigators that wanted bone marrow specimens from healthy dogs, and we were able to write a protocol where we were able to consent owners to let us collect a bone marrow sample from their pets that were undergoing elective spays or neuters. Likewise, for some of those operations, it really sort of depends upon the tissue as far as how feasible that is.